Okay, now done. Okay, show. Just 1 second. Okay, now ready. Okay, Toyo, could you start?
Okay. I talk about reusable masks by.
This is the final presentation of your whole semester, and this is kind of a conference form, and make it a little more dramatic. Not just I'm reporting my homework to you. This is reflecting, everything you have learned from this semester, and make it a little more dramatic and exciting.
Okay.
Okay, let's welcome to you.
I'll talk about the exciting interest on reusable masks by Professor Kim Il-do at KAIST. Let's start. Contents are, first contents is problems with exciting masks, and we'll talk about then nano mask differences, actually positive side, and manufacturing method and verification process, and then talk about reusable masks mechanism, and last finally commercialization we talk. And first, existing mask is made with a filter that emits polymeric materials with a melt-blown method. The method of filtering fine dust or germs by combining layers of filters by creating a static electricity. This MB method is melt-blown method is MB method's point is using static electricity, and they have a lot of problems. First one is static electricity formed on the surface of the fiber is lost over time when exposed to water or another solution. It causes the degradation of filter performance because the dust or germ can't penetrate because of mask's static electricity. And second problem is, as the fibers are randomly intertwined, the pores vary widely. It means we should to overlap several filters to make a same size pores to protect the dust or germs. And nanomasks has differences. It has right-angled or closely aligned fine nanofiber, and the thickness of the yarn is silk is much thinner at about 1/20 of the normal melt-blown filter we talked, and it causes the pore size is small and the same and thinner than the existing filter, so we can breathe easily while protect well as well as melt-blown filter. And this is manufacturing method of nano filter. And let's see the left side scheme. This is a normal filter. I think they and solution we they lab had to made a solution with polymers, nanopolymers, and they reacted on a system. So these red circles and blue one and yellow one is a such as grow mechanism. So the and the down picture is grown synthesis by TEM, and right side picture is expression by 2D graph. Actually, this is a 3D picture, and we should this axis. We should see the axis, and first growth is XZ plane growth, and second growth is X growth, and second one is Z growth, and third growth is jet growth, more jet growth. So point is they made a nanostructure on the normal yarn. And they had verification process too. This is X-ray transmit X-ray scattering on the filter, and x-axis is a theta, is a angle around the filter, and y-axis is a scattering intensity. So in the solution, they had to nanostructure growth on the filter mask 1 hour, 2 hour, 3 hour, and they grow up, and X-ray scattered more and more on the filter. It means it had better structure to protect the dust and germs. And let's talk about reusable masks. This filter is reusable after washing with soft ethanol, as we saw on the lots of newspaper, and they're on some picture on 20 washing 20 ethanol ethanol 20. Newspaper said their filter structure doesn't change enough to don't protect the germ and dust. And but actually I couldn't find the mechanism of reusable reusable masks. Lots of information said their nanostructure on right angle stacking or right angle stacking causes the stable structure on filter, so they can work. And but this good masks can cause the reduce the trash caused by lots of consumption of multiple masks, but we can't commercialize these skills because since it was developed as a new material, not a multi-bron mechanism, it takes a long time to verify and approve it by the KFDA 식약청. So on commercialization, they should take a long time. Okay, and thank you for listening.
Okay, thank you, Do-yoon. Now, guys, the questions to Do-yoon.
May I ask some question?
Yes.
Go ahead.
I saw the reusable mask used some ethanol to wash the used one to clean some dirty material. Is there any concentration standard for using those methods?
Concentration, you mean the dust or germ concentration or ethanol concentration?
I mean ethanol concentration.
I couldn't find it.
Okay, I see. How about the germs concentration you mentioned?
Actually, I couldn't find too.
Okay, thank you.
John, what was the second question? What concentration?
Dirty material, I think.
Dirty material concentration. When in the beginning, you mentioned about the normal MB filter. What is the MB filter? Or MB method?
MB filter is existing masks skills, and it is made by 전기 방사법. So filter on the filter scientists go make static electricity by 그 전기 방사법. So it protects dirty materials.
Any comparison of this new material. In the next page, you have two pictures. So yeah, next page. So here, this page looks like some flower shape with a 2 micrometer size, and 2 pages down, there's another picture. Now this one is even smaller, 1 micrometer. Now this is a kind of linear structure. What's the relationship between these two? Why the one picture shows like a flower, the other one is kind of a fiber?
This flower is on the yarn. I think this flower structure is on the yarn, and
you can compare those size with the scale bar. So according to the scale bar, this flower is about the 10 micrometer size, right? And if you go to the fiber structure, now the scale bar is 1 micrometer. The flower should be visible here, but we don't see the flower. What's going on?
This. I think this bar means resonance of.
That's a reference bar, so-called the scale bar we call. So then that's kind of reference, you can compare those size. So those diameter is about 400 nanometers, so that's approximately correct. And the flower is about 10 micrometer size. Then we should see the flower here. Why all of a sudden flower you don't see? In this case, if you see those 2 micrometer size of scale bar, then flower diameter can be easily 10 micrometer, isn't it? Or
Yes.
this too is a different material.
Actually, this flower structure is from the lab website on the nano filter to protect the dirty materials, and this I don't.
Okay, seems like this is an interesting new material. But you said this may take some time to get approval. What is the hurdle for getting approval to be in the market?
This order?
So what is the problem? Why should it take a long time? We need the mask right away.
Because mask is the very close on the face, so it is important to with long problem or other health problems, so it should conform by KFDA to being commercialization. So, and also it was developed as a new material new material. So it has the big hole.
Okay, what kind of a test should be made then? This is closer to the nose, so it should be safe. Then what is the concern? Any small particle is released from mask? What can be the concern?
One thing that I can think, this nano this nano manufacturing method is this I studied this nano flower or this nano structures is 전기 방사 on the filter, so they came out of the filter surface and invade the our lung.
Okay,
Actually,
any other question to Doyun? Seems like this paper was published in 2018, 2 years ago. But the product is not in the market yet.
Yes.
You may mean no, right? No, it's not. Or yes, it is. Okay,
no.
thank you, Doyun.
Okay.
So small English the lesson here. So let's just say if this is an interesting work, and I'm interested or I'm interesting in this work. This work is interesting or this work is interested.
Interesting.
This work is interesting, and I'm interesting or I'm interested.
Interested.
I am interested. If you say I'm interesting, then I'm funny guy. I am interesting. Material is interesting, and I am interested. Okay, so some of you had a little hesitation to make that choice. So small English lesson. So let's move on. So second speaker is Dongwoo, and Dongwoo is going to talk about how COVID-19 kills.
Okay, so can you see my screen?
Yes.
Okay, so then I'll start. So to start, before I start, my presentation is not actually like a very 100% scientific presentation, but like it's kind of concentrated on the medical facts and the symptoms and how the patients are suffering from the disease. So I will first have to mention that, but still. I tried to provide a general view of what we know about how we are contracting and fighting with the COVID disease, and what we know and what we have to prepare for it. So I'll proceed. It's gonna be really long. So an hour before this presentation, I've looked up the chart, and it says that there are more than 8 million cases and 400,000 people have already died. So you can see that the daily cases are increasing every day, even though it's already June. But the daily deaths are decreasing. So I guess any health, the peak is over, but you never know. So my contents will be mostly focusing on the 3 cohort studies in Wuhan, China, and in New York City and in London. So I'll discuss about 3 papers that are published by the hospital hospital experts in these three cities and what they have assumed that is the most threatened risk factor for each of the patients in there. So after discussing about that, I'll proceed to what the additional impacts of the COVID disease might be and how we're fighting with it. And to overcome it, I'll give my own proposal of how to fight it in the end. So to start, I have to first mention that not all the people who contract the disease are ill. So if we are, let's assume we are tested positive, it means that 70% of us are asymptomatic or doesn't have any signs of fever or cough or anything like that. And those people get into quarantine in their own homes. However, the next 30% are symptomatic, which means they show symptoms. And of them, 15% of them have severe disease and have to be admitted into the hospital, while the 80% of them are not. So this means that only like a 5% of those who get the disease are ill. However, as we see in the chart in the front, it's still killing many patients. So we have to take a profound research on how they react on these hospital admitted patients. By the way, this chart is taken from the British survey, so it only takes results from British citizens, so it might be different in Korea. So I'll focus on the cohort studies taken by the scientists. And this cohort basically means that the scientists shown in here are in a hospital and conduct and research about the various symptoms that the patients have in that hospital. And they could show either severe symptoms of COVID or not. And they also want to know what the patients have been previously exposed. So they could either have various symptoms like smoking or diabetes or the other diseases. So scientists calculate all that stuff, and taking that into account, they make a result on which shows that if you have some diabetes or like old age, you have the more highly percentage of to contract the disease. So this is the meaning of the cohort study. So I basically gathered data from March, April, and May from 3 different countries, and also various newspapers that show how COVID killed. So the first is the research paper that has been come out in March by hospital officials in Wuhan. So this paper is quite famous. It has almost more than 3,000 citations, even though it's been published in March. So you would like, if you're interested, you would like to search this article up. So the general impacts shown in here says that the COVID impacts these all organs in various people. So it could either go from our brain to the intestines, our lungs, and to our kidneys, and like all of them. So you can see that the most common things are cough and shortness of breath, but there are other various things also implied. So we'll take a look on that. So the first study I would like to focus on is the cohort study in Wuhan. So this is basically a paper which mentions patients that have been admitted to 2 different hospitals until January 31st. So it's a very early clinical data. So there are 191 patients who have been admitted, and among them, about 50 people have died. So that's a lot of percentage. And this is the symptoms that the survivors and the non-survivors have each shown. So these patients have all died when they have reached about 19 days after admittal. And the one interesting thing that we have to focus on is that we could find sepsis, which is an infection to our body by the virus, and ARDS, which means that which means acute respiratory disease syndrome. So these two symptoms has been found in 95% of non-survivors. So this means that ARDS is the major cause of death. And the research office officials have also tried to find what's the risk factors of the disease. So they first found out that old age is a major cause. So you can see here that from the non-survivors, the mean average age is 69. However, in the survivors, it's only 52. So increasing odds of death is caused by an older age. And the second risk factor is a high SOFA score. And the SOFA score means how our body is damaged. So it means sequential organ failure assessment. So this basically is a value which means how you're feeling bad. It basically means how you're being infected by the disease. And also, one interesting thing about this research is that if you have a high D-dimer level, you're more likely to die. And you might as well know what exactly is this D-dimer. It's actually a protein product of cross-linked fibrin degradation. And it basically means that if you have a blood clot or your blood is kind of mixed together in your body, then you're likely to have them degraded by immune responses. And when the immune responses are taking place, those garbages that are left after they clean up your clot is basically called a D-dimer. So you can see that non-survivors have very high D-dimer levels before they die, while the survivors have basically none of them. So if you have a blood clot, you're more likely to die. So this is the result in Wuhan, and we'll proceed to London and New York. So the research taken in UK was a very broad clinical trial with 20,000 patients from 208 hospitals. Among them, 41% were alive, but 26% of them died. So that's still a very high amount. And the symptoms shown by them are, as you know, fever and SOB and cough are majority shown in these patients. And also, we would like to take into account that most of these patients have comorbidities, which means 합병증. So they already had diseases before they contracted the disease, which means that you're more likely to get the disease if you have one of these chronic heart disease or lung disease or diabetes. So we wonder why is that? Why is the reason of that? So before I tell you the reason, I'll also have to mention the studies in New York, which also investigated 5,000 patients with infection from March to April. And among them, this is quite complicated, but you can see that among them, like 36% of people who are admitted to hospital were experiencing critical illness, and among them, like more than half of them went to a hospice or had a high likely chance to die. So in this graph, we can see that we have a different. This purple graph is basically the percentage of people who have died and who are alive, and the yellow graph means the people who have survived. Sorry. The purple means they're alive, and the yellow means they're died. So you can see that in male, this difference is quite short than females. So you can see that male are more likely to die when they have the COVID, but still, it's not much of a difference. You can see that it's not quite different. So, and this is about diabetes. So when they have diabetes, they are more likely to live, but when they have a diabetes, this difference between two functions are low. So you can see that you're more likely to die when you have one of these diseases. However, as shown in Wuhan, the New York study also shows that the D dimer is really important. So the D dimer is a measure of how your body is responding to inflammation. So if you have more inflammation when you first enter the hospital, you're more likely to die. Then the other various impacts like diabetes or obesity or old age, that doesn't really matter. But inflammation is really important in tracking your symptoms to really tell you that you're gonna die or not. So as shown in here, I'll not talk about the differences, but the similarities of these results. You can see that we have a high rate of critical illness where we have high D dimer levels or low oxygen levels, and also these various comorbidities. So we're going to see why these three reasons contributes to the reason of death. So this is basically because the COVID COVID binds to the ACE2 receptor. And as you all know, ACE2 is a receptor that resides in our lungs. And it also has an anti-inflammatory effect. So this means that people who are old and have diabetes and hypertension have more ACE2 in their cells because they are prone to have more inflammation in their body. And to suppress that, we need ACE2. So when SARS-CoV-2 goes into our body and kills this ACE2, our inflammation is going to get worse. And also, the location of ACE2 is on epithelial cells, which supply oxygen to blood. So if this doesn't work right, that means that the epithelial cells are not going to work right, which means that the capillaries or 보세혈관 in our body are going to burst. So when this happens, blood enters our lungs, and because of blood entering the place they shouldn't have to be, they get clotted by immune responses, and therefore D dimer level increases. So this is the damage on our epithelial cells, and we also have to talk about how the ACE2 reacts to control our body. So ACE2, its main function is to regulate the nitrogen oxide levels. And as you all know, nitrogen oxide makes our blood vessels to constrict or dilate. So if this is disrupted, our blood vessels are out of control. So this means that we have lower oxygen levels and we have a damaged lung. So all these effects take into account to make our body more vulnerable to infection and to have an ARDS, or acute respiratory disease syndrome. And also, we are prone to have more inflammation. So this also leads to cytokine release syndrome that we have talked about. So I've highlighted ARDS in green because this is the major cause, like 95 or 99% of the reason why patients die because of COVID-19. So you have to memorize these 4 words. So this is like the summary of how the COVID enters, and this makes our body to have inflammation and to cause death. So you can see that in here, when we have a disease state, we have a high chance to have vascular leakage and to have coagulation and inflammation. So moving on. I would like to also talk about the additional impacts, not only about how the lungs and our inflammatory responses are damaged, but I guess because the time is not right for me to discuss all of this stuff, I'll skip these. And but I also have to mention that when most people who have severe COVID-19 are taken into ICUs or 음압병실. So when they get intensive care in these beds, they are likely to have poor muscle strength and poor lung function after they get treatment. And people who have ARDS get it worse. So if you're basically sitting on a hospital bed for a few months without doing anything, so it's hardly best to imagine you will have various impaired functions on your brain and your muscular activities. So people mostly suffer from anxiety and PTSD. And yeah, this is also a severe problem that we also have to take into account. So people who are fighting COVID. So if you're contracted with COVID and taken into hospital, the doctors will give you extra oxygen and give fluids to prevent you from dehydration. So this is the major two things that the nurses are going to do to you. So they'll stick a nasal cannula, a plastic tube, to provide with you with extra oxygen because the ACE2 is damaged. And because dehydration thickens the blood, and blood thickening is a major cause that causes blood clotting, it's very important to get ourselves hydrated when we have the COVID. So these two are important, and also it's best to have a good night rest and to be on ventilators if you have a suffering of breathing. Also, let's skip please. And also, there are many clinical trials that are taking place into fighting COVID. So if you look up clinical trials in certain website, you can see that there are almost 2,000 clinical studies that are taking place in various hospitals. So we can also take a help of them, even though we're not sure they're going to work or not. Also, before I end this presentation, I would like to propose some things that we could take into account. So what I'm suggesting is that first, the testing kits should only test whether or we are positive or not, but it should also test if we have a high level of D-dimers or we have a high level of inflammation. Because if we test these two, then we could know that we are going to have a severe symptoms or not with a high accuracy. So with this data, clinical faculties could take a better care of us, like sort of like a precision medicine. And also, clinical trials are needed to find usage of anti-thrombotic drugs. So this is the drugs that can help us from reducing the blood clots. So if we have severe symptoms, patients of COVID, we could use them to degrade those blood clots and to increase the survival rate. But it's just only a suggestion, so I don't know if it'll work or not. And also, the deep, profound study of SARS-CoV-2 and ACE2 is also needed to find these various stuff for accurate drug development in clinical trials. And I believe that many scientists are doing it right now, and many research papers have been published. And also, finally, government aid has to be made for survived ICU patients because, as I said before, if patients suffer from ARDS, they're likely to not be in work or to do normal jobs after they go out into the society after they've been fully treated. So I guess government subsidies or kind of that are needed for them to revive. So this is the end of my presentation, and thank you for listening.
Okay, thank you very much, Tomo. This is the very good the presentation you did. Your good summary of different the references combined together and made the good presentation. But one small other comment is, there are some abbreviations you have used. It's better you give those abbreviations for every pages that you show up. For example, ARDS, CRS, PTSD, or even the nitric oxide or NO. So this kind of thing So to the familiar people, it's okay. But if you just hear the talks for the first time, there are too much information at the same time. Okay. So almost every page is if you have that word, even though you mentioned the verbally before, it's better to give that the full name for each pages. Then it will help to grab the attention. Okay. You gave a lot of information, but if you summarize, what kills the COVID kills how?
So we could take two approaches. So if we could first look at the dead patients and see what causes the death, and we could measure all the patients and see what most makes those all patients to die. So if we look at the dead people, we could see that ARDS is the major cause of death.
So what is ARDS?
Yeah. And ARDS is the acute respiratory disease syndrome. Oh, okay, I'll show
So
you
that's
here.
kind of the first the symptom we recognize, kind of the respiratory the syndrome, like the SARS. That's number one. That's obvious first choice. Then what is the other, or the unknown, or the new, or the discovery?
Discoveries.
Lung inflammation is the number one, of course. And what else did you find?
We could also say some things about our kidney failure. So I
Kidney
didn't
failure.
mention it in my
Okay, kidney
presentation,
failure.
but some patients, yes.
Okay,
And also malfunctioning of our heart. So we have heart attacks and seizures.
Okay, heart problem.
And also we have to mention CRS, I guess.
What is a CRS?
Cytokine release syndrome. Yes.
Say again.
This is also a cytokine release syndrome. Is it correct?
Cytokine release syndrome. So this means this is a kind of an over-immune response, correct? So it's kind of a cytokine at the storm, what you call. So it secretes too much cytokine, and immune system was over-boosted, and then it generated some problem. So that's a CRS, cytokine release syndrome. And you also mentioned something like a blood clotting.
Yes, but blood clotting eventually leads to ARDS. So like this is the first reason why they get contracted with severe diseases and eventually lead them to death.
So that means that the blood clogging eventually affect the lung as a major the damage site. Eventually, yes. You said this is related to the ARDS. So that means blood clogging is eventually connected to lung syndrome.
Yes, because if our blood is clotted, we are hard to ship our oxygen to our one body part to another. So that eventually leads to low oxygen levels. So
is that the oxygen delivery problem? That's kind of a systemic problem. But the other way to see is the function of the lung itself, right? So both of them yes. Can affect the oxygen delivery. So question is, blood clogging is directly affect the lung function, or that's just adding up the problem for the oxygen delivery? Let me rephrase the question. Whether this blood clogging is directly affecting the lung function? No, it's parallel.
I think I have to research on about that. I found about few research papers on that, but I don't exactly remember how are the correlation between them. So.
Okay. On this page, you also mentioned the NO somewhere, the regulating NO. Regulating means it's a decreasing NO or increasing NO?
Both of them. So if they're
okay. So then NO's function is NO is a nitric oxide. NO's function is a dilation of the vesicle, right?
Yes.
Then if you dilate the vessel, does it help or does it help inflammation or block inflammation?
It helps inflammation because if we dilate our blood cells, more oxygen are likely to pass through. So
Then inflammation is the increase or the decrease? With the
uh,
dilated blood vessel, more oxygen supply, inflammation
yes.
increase or decrease?
So inflammation decreases.
Okay. So this kind of a concept is hidden here and there. So there are a lot of information here. So after this, the kind of a lot of information at the presentation, it would be good to have some summary pages. Then
Okay.
we just heard this is a kind of a lung inflammation, perium, but actually it has a different potential possibilities. Even you mentioned something about the diabetics. So those can be either cause or other important factors for the affecting the COVID patient. So that kind of a summary would be quite helpful. Would you add up the summary pages and send me that file again?
Okay. I will.
Okay, and also add those all abbreviation, the full the name, then it would be helpful.
Okay.
Okay. Okay, any question to Dongho? So today we have 3 presentations, so we have a little time. So we could take a little longer, the version like this. But Thursday we have 4 talks. So everybody should be at to keep your time within 20 minutes, okay, including Q&A. Okay. So then let's move to the third talk. Third talk is by Juhan, and he is going to talk about the coronavirus mutation as its impact.
Is there the screen sharing right
Very
now?
good.
Thank you. Hello everyone. I'm going to this is the last day, last presentation of this semester. So to begin, I was wondering to explain about the specific mechanism of the mutation of COVID-19 in these days, just like the professor mentioned in Thursday, like A, B, and C in these days. But actually, there is no tight mechanism research result. So I unfortunately I will going to presentate about some just kinds of the symptom and the difference between the mutation of COVID-19. So and this presentation will be the constant line, parallel line as the Dongwoo did just before. So just let you guys know to take the memory of the things he said just before, just like the mechanism of the COVID-19 in the lung, and listen to my presentation, please. So to begin, we all of us may know that the bat is quite related to COVID-19's release to human, and this is the genetic tree of representative COVID-19. This can you can see the bat SL blah 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 is very related to SARS-CoV-2 or COVID-19, and you may can also see SARS and MERS. MERS can is also contained in beta coronavirus as well. So scientists had take the difference between those kinds of disease and found out that some of the genetic materials are difference between those original one, and they define those things as the mutation type of the coronavirus. I will tell you the detail of it. So this is the map that the coronavirus, COVID-19 is spreading over the world. And you may can see there is some kinds of relationship between the types there that is quite big in those countries, and is very related to those regions where those countries here. The China has mostly type B and A, and while the European area has B and B, and the North America and South America has type A as well. So uh, those scientists have very big question about why those kind of reason is very became very critical to those coronavirus type, and they finally figured out that the virus may be constantly mutated to overcome the different level of the immune system in our body. So they tried to overcome our immune system to attack our lung, as the Dongwoo mentioned, and uh, those kinds of the mechanism in our immune system is quite has some difference between related to our the region here. So this means the Asian people and South Africa people have quite some difference mechanism, or the protein is some has some significant difference between those people. So that is not a quite definition of this mechanism, but uh, a lot of scientists believe that coronavirus have this specific characteristic. So you should you may not right good. You may not believe those kind of theory, but this could be one of the way to relate those research topics as well. And this is the details that coronavirus COVID-19 A, B, C types has mutated by the time. You can see the picture in the middle, and the type A virus is the most related form as the bad test in the first period. The type A is the closest form in bats, and this is considered as the root of the corona pandemics has begun. So there have been two kinds of subcluster mutation, and the type B is the mutation version of A. So as A is more related to bats or animals, but B is more related to Wuhan person. So you may saw the in this map there are mostly type B in China. So B is more B is more the related form to human, while A is not that deeply attached to our human, our body. And some scientists have found few samples that there is few mutation in T3A2C and C281444T. That is genetic material site of that the ACE2 that attacks to ACE2. So those kinds of mutation or difference make the difference between type B and type C. So while type B is related to Wuhan, type C is quite efficient, quite significantly deadly, affected deadly to Singapore or European as well. And there is another definition of the COVID-19 mutation form by phenotype. You may relate it to L type and H type as low and high, because type L is just quite not have quite big significant quite big symptom in our body, while type H has very severe ARDS symptom as well. So I mentioned the ARDS as acute respiratory blah blah blah. So type L has I mentioned those kind of symptoms as arrow. The elastance and VA that Q ratio is very low, which you can just check it that the patient and the normal person is very quite has not that much significant difference between them. So that is why the 80% of patients can be stay at home, not to the hospital at Dongwoo's presentation. And type L could be have such some small local subplantar interstitial edema, but that is not related to whole lungs disease. While type H has severe symptom, so it can damage to the whole lung. I will show you the CT picture in next slide. That is the CT shot in of the L type patient and H type patient, and you can see that there is a white material in picture B, while A picture is quite clean. So this is the white material is 염증. So 염증이래.
Inflammation.
Inflammation blocks the air oxygen in air to go into the pulmonary vein. So that cause the oxygen pressure in our blood very significantly low. So the L type patients have about 95 value in PaO2 FiO2. This is the method that to check the SOFA score, while the H type patient has 84 in PaO2 FiO2. So that means that is the symptoms explanation of the lung, that those lung cannot transport the oxygen from the air to our body. So those kind of phenotype can be identified by CT scans, and some of the computer science scientists are developing the algorithm to differentiate by the CT scan image. Thank you for listening.
Okay, thank you, Juan. So questions to Juan?
So, is there any kind of biological or scientific marker to separate type A, B, and C coronavirus? I mean, the
Actually,
definition sounds quite ambiguous?
Actually, I searched the markers as you mentioned about 2 or 3 days, but I could not find those markers without without.
Now, what can be the current criteria of distinguishing of this the A type, B type, G type?
I mentioned the two site is mutated at the genetic material. I cannot find which genetic material is mutated, but those kind of difference can be the markers.
Difference in
The mutation of the genetic material in protein, not protein, but
John, you implied something, but you didn't give that the critical word. Is it protein level?
I think I should check it to those paper.
Okay, when Sejin mentioned about the biomarker, that can implies most likely there's a protein level of difference. So current A type, B type, C type is based on the sequence of RNAs. So current the classification is based on the RNA sequence. Then mutation also means that the RNA the sequence at the level of mutation. Now, if the study is developed further, then different sequence of the RNA, they can make a different protein potentially, and the protein can be recognized by different antibody. Then we call this difference as a serotype. Sero means it's a serum from the blood. So that means this antibody can recognize those differences. So it seems like this is still quite early at the stage. I don't think it's developed that far. Based on antibody-based, the differentiation is possible. It's not clear. It takes a longer time. So for now, the general the classification is made by the sequencing. Now, interesting the point Juan suggested. This B-type generation or the deriving from A-type is during those adaptation in the patient. It implies that from same A, when the B is made, does that mean that this B can be independently generated in different area? Okay, let me clarify again. Usually when we have this kind of a sequence, that we assume that A is divided or the branched out into B, and then B is changed into C. So can you go back to the one slide back? Then by looking at these sequences, A is yellow and B is orange and C is red. So based on this sequence difference, we believe that we can see those transferring track. So A type is the original one, and then it has some mutations made in Wuhan area. So that's why they have both A and B. And maybe some of them was moved to America. And if you look at those C is Hong Kong, Singapore, and some part of Italy. Then this C may be generated from B. Then it's not directly transferred from Wuhan area. It was maybe from North Canada or other part of the Europe or South America or something like that. So with this sequence branching out and also the period when this appeared, then we can track those traveling of this the virus to different area. That's kind of assumption. So if you follow that model, then Singapore and Hong Kong should be kind of a little later stage, and they had different branches. Maybe from Italy they were back imported, or it was mutated in Hong Kong and then transferred to Singapore, and from Singapore to Italy. That's kind of a common model we think. But Juan implied something like that. This mutation is generated by during those adaptation, the fighting against our immune system. Does that imply that independently different B can be generated from different area. Is that what you meant, John? You said something like believe or not. So is that what you meant?
Yes, because there's not that much scientific belief about those kind of A and B and C type differentiation. So I cannot say that this is the quite a fact here, but that is mostly belief types of model in scientific society. So I present it just like that.
Okay. Okay, other question, which one? Okay, if not, so thank you all the three presenters for today. So as I said, on Thursday we have 4 presenters. So everybody be ready with a little more succinct the presentation. So every the presentation will be within 20 minutes. Then we cannot be too late. So how many of you are currently in the campus?
I'm in the campus.
So Sejin is in the campus. How about others? You are at
I'm
home
also-
or you are in the campus?
I will go to campus at Sunday.
Someday means sometime this week.
Yes, in this week.
You have the experimental class in the campus.
Yes.
Okay. Anybody else coming to
I'm
campus?
in campus now.
Who? Doyon. Okay.
Me.
And
To
someone you are coming to?
yes.
When do you
Actually,
come? When do you plan to come?
actually now I can go to school right now.
You are in Pohang?
Yes.
Okay. Then Juan, Younghoon, and Dongwoo is at home.
Yes.
Professor.
No plan to come this week, next week. Okay. Then maybe we should take the final exam as scheduled, the online, if you don't plan to come. Okay, guys, thank you and see you on Thursday. On time, we should start on time and we should keep the time, then we can finish on time. Okay?
Thank you, Professor.
Okay.